Transpiration and stomata are closely linked in plant physiology, playing crucial roles in water regulation, gas exchange, and overall plant health. Understanding these processes and structures provides insight into how plants manage their water needs and interact with their environment. Transpiration is the process by which water absorbed by plant roots is transported through the plant and then evaporated from aerial parts, primarily the leaves. This movement of water serves several essential functions. Cooling the plant. Transpiration helps cool the plant as water evaporates from the leaf surface, similar to how sweating cools animals. Nutrient transport. The upward flow of water from roots to leaves transports dissolved minerals and nutrients essential for plant growth. Maintaining turgor pressure. The continuous movement of water helps maintain turgor pressure within plant cells, which is vital for maintaining cell structure and function. Driving force. The cohesion tension theory explains how transpiration creates a negative pressure, tension, in the leaf's air spaces, pulling water up from the roots through the xylem vessels. Stomata, singular stoma, are small openings on the surfaces of leaves and stems that facilitate gas exchange and regulate transpiration. These pores are flanked by two specialized cells, known as guard cells, which control their opening and closing. Structure and function of stomata. Guard cells. Guard cells are kidney-shaped cells that swell or shrink to open or close the stomatal pore. They contain chloroplasts and can photosynthesize. Stomatal pore. The pore itself allows for the exchange of gases, CO2, O2, and the release of water vapor during transpiration. Regulation of stomatal opening and closing. Light. Light stimulates the opening of stomata to allow CO2 to enter for photosynthesis. Blue light, in particular, is highly effective at triggering this response. Carbon dioxide. Concentration. High internal CO2 levels cause stomata to close, while low CO2 levels prompt them to open, optimizing photosynthesis. Water availability. When water is scarce, guard cells lose turgor pressure and close the stomata to reduce water loss. Hormonal control. Abscisic acid, ABA, is a hormone that signals guard cells to close stomata during water stress, helping to conserve water. Mechanism of stomatal movement. The opening and closing of stomata involve changes in turgor pressure within the guard cells. When guard cells take up water, they swell and the pore opens. When they lose water, they shrink and the pore closes. Ion transport, mainly potassium ions, into and out of guard cells influences water movement. Uptake of potassium ions into guard cells lowers the water potential, causing water to enter and the cells to swell, opening the stomata. The relationship between transpiration and stomata is intrinsic to plant water regulation and gas exchange. The following points highlight their interaction. Water loss regulation. Stomata control the rate of transpiration by opening and closing. This regulation is vital for maintaining water balance within the plant. During hot, dry conditions, stomata close to conserve water, reducing transpiration rates. Gas exchange. Stomata enable the uptake of CO2 necessary for photosynthesis while allowing the release of oxygen. The balance between water conservation and CO2 uptake is critical. Closed stomata prevent CO2 entry, reducing photosynthesis, but open stomata increase water loss. Environmental responses. Plants respond to environmental conditions by adjusting stomatal behavior. For instance, in high light and low humidity, stomata may partially close to balance water loss with CO2 uptake needs. Transpiration stream. The continuous stream of water from roots to leaves, driven by transpiration, also helps in nutrient transport. Minerals dissolved in water are carried along the transpiration stream and distributed throughout the plant. Transpiration and stomata are central to plant water management and overall health. Transpiration facilitates cooling, nutrient transport, and turgor pressure maintenance, while stomata regulate water loss and gas exchange. The dynamic regulation of a stomata in response to environmental cues ensures that plants can optimize their physiological processes, balancing the need for CO2 for photosynthesis with the necessity of conserving water. Understanding these mechanisms provides valuable insights into plant adaptation and survival strategies in varying environments.